Hey, hello everyone. Yeah, thanks for inviting me to talk today. I'm Matt Coelho from the Wellcome Sanger Institute in Cambridge, UK, and I've just got funding for my own lab there, so from Cancer Research UK, and I'll be looking at using gene editing tools to understand cancer variant function and how this impacts on therapy response. Um, and so, yeah, the cancer variant function problem is a somatic mutation issue, and there's actually very little data on post-treatment biopsies from cancer genome sequencing because they're very hard to come by, these cancer genome biopsies, and actually understanding what the variants do in terms of therapy response often is descriptive and requires functional validation on a variant-by-variant basis. So to do this in a more systematic way and less retrospectively, we can do this using gene editing. And so this approach allows the direct coupling, of course, of the variant, the genotype to the phenotype in the context of therapy. So we can analyze this in the dish and hopefully recapitulate what's happening in a patient which relapses on therapy. And this can be very impactful because many cancer patients who relapse on therapy, they don't have a clear second-line therapy which will be effective for that variant, for example. So today I'll mostly talk about the base editing strategy for several reasons, and John nicely introduced that this morning, so I won't go into too much detail, but essentially we're choosing that for cancer over these very complementary other gene editing technologies at the endogenous level, because base editing is highly efficient in several different cancer cell contexts. And that's important when you're looking at therapy response, because we need to choose a cancer cell model that responds to a certain drug. So you need to have that flexibility there. And also, many of the cancer cell models we work with have many copies of the oncogene. Some of them are aneuploid, and therefore you need really high editing efficiency to get strong phenotypes. And so another reason why we might use base editing in cancer is actually it uses, for example, the cytidine base editor uses these deaminases, which introduces C to T mutations. And actually in cancer, these ApoBec enzymes are highly dysregulated, meaning that we can recapitulate quite a lot of clinical variation, which is seen in the cancer genome using these base editors. And just to show you in a toy system in these reporters, so this is actually a transgene that we're base editing to look at the activity of the base editor, which we've called BE flare. This C to T mutation at codon 66 in BFP can be changed into a tyrosine, and that can track base editing activity by a GFP transition here. And so this single nucleotide change can allow you to, 1, track base edited cells, 2, test the activity of several different base editors, new base editors, different base editor architectures, and also enrich by FACS for actively base edited cells, for example, a second endogenous locus. And so these are the kind of cancer cell lines which we use, the colorectal cancer cell line with an inducible base editor, here the cytidine base editor, which is turned on with doxycycline, and you can see in this FACS plot that we get this BFP to GFP transition here. So this is just a transgene, and one of the tools we use to make these cancer cell models, which can do base editing, but now we want to do this instead of a transgene at the endogenous level, and actually at a pathway level, because many of the pathways we're interested in, it's not just a single gene, we want to look at many signaling proteins and modules to understand how that's affected in response to therapy and in cancer signaling when it goes wrong. So one example I'll show you here, a pathway level mutagenesis, semi-saturating mutagenesis with base editing is on the interferon gamma pathway, and we're very interested in that because not only is it interesting thus in inflammatory disorders, but also leukemias, but actually anti-tumor immunity. And so many cancers are treated with immunotherapies very effectively, but there are occasionally mutations in this interferon gamma pathway, which is shown here on the right, which can affect anti-tumor immunity and actually cause resistance to immune checkpoint blockade or T cells. And so here we were able to functionally characterize several variants of unknown significance in this pathway, and also using cytidine adenine base editors, validate them in more complex models, which goes to my previous point, in organoids, so tumor organoids, where we can co-culture them in a complex system with anti-tumor T cells from the same patient, and we were able to verify that some of these mutations actually cause resistance or sensitivity to anti-tumor T cells. So this is what some of the base editing data would look like for this kinase downstream of the interferon gamma receptor, JAK1. And so here is the amino acid position of the kinase JAK1, very similar to the TYK2, which we saw earlier in the conference. And we have the Z-score here. So any mutation, so these are each guide RNA installing a different variant. Each mutation here, which is loss of function, is actually causing resistance to interferon gamma in the cell. The cells grow faster, and they are loss of function. And so we're seeing some missense variants, which are as potent as splice variants and nonsense mutations. And actually, we find these rare gain of function mutations, so dropout, which have been found in leukemia as drivers, but also some myeloproliferative disorders. And here there's a nice cluster, which I'll show you in a moment, which map to a protein-protein interaction interface. But we're really quite excited to see that some of these missense variants, which didn't know what they were doing before, have been found in patient biopsies which have failed to respond to cancer immunotherapy. So the gain-of-function cluster, which we identified in this crystal structure of JAK1, actually, it's stopped rotating here, but yeah, there's a protein-protein interaction interface with the negative regulator SOX1. So hopefully we can exemplify here that we can identify important functional domains in proteins, which might be drug targets. In blue is the kinase domain, which we identified. So the kinase active site, which we identified with these blue missense mutations, which are loss of function as well. So this could help drug discovery campaigns, not just looking at drug resistance. And we recently published with Matthew Garnett's lab a couple of weeks ago with GlaxoSmithKline, actually functionally looking with base editing at their drug target WRN in cancer to see which parts of the protein are important for cancer cell function and cancer cell survival. And that directed the drug discovery. So that was all proliferation screening. I'd like to also talk about more high content readouts of single nucleotide variants, which we're just following on nicely from the last talk. And so we actually coupled this base editing screen of JAK1 to, not proliferation here, but single cell RNA sequencing. And so this is 10x data. And in this case, we have a barcode in the guide RNA, and we read that out, and actually each clonal population of cells should be genetically identical because they're from the same daughter cell. So this is this UMAP here showing that looks quite binary, but I'll show you in a moment it's not quite that binary. So we have these wild-type-like cells on the right, and then these cells which don't respond to interferon gamma transcriptionally. But actually, we wanted to clean this up with a more unambiguous assignment of the genotype by mapping the 10x data to single-cell DNA sequencing. So we can unambiguously assign by these barcodes the genotype and the transcriptome, the phenotype. And obviously, we've seen that with guide RNA, sometimes they don't work. So this actually takes away that level of uncertainty. And that's in this bottom plot here. So here we can see now we've introduced missense variants, which we know are there from the DNA sequencing, and they are benign because, functionally benign because they're not really doing anything to the transcriptome in this assay. So it's not so binary, and hopefully this shows you a little bit about the more nuanced information you can get from the function from these high content readouts. Because we find each row here is a different variant, and each histogram here is a cell population and the transcriptional impact. So we can see here that actually the wild-type cells and the loss-of-function cells are very transcriptionally different for the loss-of-function and the wild-type. But here is an intermediate population of variants, which we've called here separation of function variants. And you get a cellular transcriptional heterogeneity. So some of the cells with the same variant, which we know is installed because of the DNA sequencing, some of them are in this wild-type-like state, and some of them occupying a loss-of-function-like state. And we think that's to do with the expression of this regulator IRF9 of the pathway. So this is giving us a bit more information that we never would have seen with the proliferation screen. So I'd just like to finish with some unpublished data here with applying all of these approaches to look at not response to immunotherapy, but response to targeted agents in cancer. So these are molecularly targeted drugs, and we'd like to prospectively map variants which change drug sensitivity. And so here we did screening with adenine and cytidine-based editors of 11 different cancer drug targets with 10 different cancer drugs, and looked to see which ones changed the drug response, essentially. We also did some single cell data at the end. And I'd just like to show you what some of these data look like for a MEK inhibitor, trametinib. So each dot, again, is a different variant. This is the growth of the cells in the absence of drug, and on the y-axis is in the presence of drug. So anything off the diagonal is something which is a drug sensitizing or mutation which causes resistance. And really interestingly, we found 4 different functional classes of variants which affect drug response. So these are the ones which map to the active site and the drug binding pocket, so they don't really have an effect in the absence of drug. These drivers cause proliferation in the absence and presence of drug, and their gain-of-function mutations. We also found these sensitizing mutations which indicate combination treatment. So in this case, an EGFR inhibitor and a MEK inhibitor would kill the cells more. And more interestingly, these peculiar drug addiction mutations which cause the cells to die in the absence of drug, but actually are resistant in the presence of drug. And we think they're actually overactivating the pathway. So that's just some data to show this is the drug binding pocket, so that makes sense. And also the drug addiction variants, as soon as you take away the drug in black, the cells stop growing. So they're basically causing this oncogene-induced senescence phenotype, and we've got some other data to show that. So we can do that on a proliferation basis, but then we validated some of these guides using single-cell RNA sequencing with a perturb-seq approach. And you can see here that actually quite satisfyingly, we're seeing the same clustering by phenotype with the proliferation screens that we see with the transcriptomics. So the drug addiction mutations are clustering with this pathway overactivation in the MAP kinase and EGFR signaling. And actually that's seen here in this histogram. So the drug addiction mutations are like the canonical drug resistance mutations, just more extreme in terms of their transcriptional impact. But we also saw some really interesting things we didn't expect, like downregulation of certain pathways, which we're looking into now and seeing if we can use these as acquired vulnerabilities. So that just leads me to summarize. We looked at lots of variants, and I just wanted to show you like the whole map of this prospective landscape of genes and variants which affect drug response, and highlight a few things where we can actually, if a patient has a certain variant, we can actually say perhaps they'd respond if you had a driver mutation in PI3 kinase to this inhibitor which we tested and is sensitive in our assays. But if you have a driver downstream in AKT, you're likely to not respond to that drug. So we hope to use all this data together to hopefully use it as biomarkers and help stratify patients if they fail on a certain drug. And yeah, so essentially we can use these gene editing tools to look at what variant functions do and how they affect therapy response. And we hope to use this prospectively in the future and not be so reactionary, work with drug companies in order to make these maps before they put the drugs into patients. And hopefully patients will be able to be treated with effective second-line drugs in the future. And that leads me to thank my previous PI, Matthew Garnett, and his group. And we've had lots of excellent collaborations on the single-cell RNA sequencing with Andrew, Leo, John, and the organoid data with Emil. And thank you all for listening. Welcome any questions.
We have time for one question. So who's got one? Okay, I will ask a question then. Okay. The separation of function variants you showed are really interesting. I guess I have sort of 2 thoughts about them. One, it sounds like you do maybe understand what's going on. And so I'd love to just hear a little bit more about that because I didn't read that deeply in the paper. And then secondly, do you worry about the accrual of like compensatory variants for some types of variants that might lead to that type of separation of function phenotype without actually having a driver you can pick up in the transcriptome necessarily that's relevant.
Yeah, so I think, so with the compensatory mutations, I think the assays are quite short, so there could be some clones of cells which have, different background mutations. So we could try to use clonal populations in the future to make that a bit cleaner, but we haven't done, whole genome sequencing. It's simply a targeted amplicon sequencing of the single cell. So that's a very good point. We think so we did some differential gene expression analysis of the separation of function versus the other ones and found that they are expressing a significantly different level of this transcription factor, IRF9. So what we think is happening is that with those JAK1 variants, they actually have just a lower or higher level of that transcription regulator, and they're occupying that intermediate state. And if you do the proliferation screen, actually, that kind of pans out over time and it accumulates over time. But if you take a transcriptional snapshot, you can identify single cells which are occupying both states. So that was an unexpected finding. But I don't think we're fully there with understanding what's going on.
Super cool. Thank you so much.
Thank you.